Our presentation is the transformation of E. coli cells into green lanterns using the green fluorescent protein gene. So a little introduction, what is transformation? It is the basic idea that it incorporates a foreign DNA into the cell. And for this experiment, the foreign DNA actually alters the genetic makeup of the cells, so it incorporates itself into the cell's genome, aka genetic transformation. And for this experiment, we transformed the cells E. coli. So the plasmid that we use to transform the cells is called P-green. The P-green plasmid contains two genes. The first is the AMP-R gene, which is ampicillin resistance gene, and it kind of does exactly what it sounds like. It allows the cell to be resistant to the ampicillin antibiotic, so it can live in places that has that, or when it comes in contact to that antibiotic, it can resist it and continue living. And then the second gene, the GFP, or the Green Fluorescent Protein, it kind of does exactly as it sounds. It allows the cell to glow under fluorescent lighting or even normal lighting in like a greenish yellow kind of color. So it makes it easier for us to identify the cell when we used it in the lab. The variable that we tested for this lab was using a different salt than what was used in our original experiment. The salt that we used was the magnesium chloride instead of the calcium chloride salt. The magnesium chloride role in this experiment was to treat the cells and to make the walls permeable, which would allow the plasmid to easily pass through the cell wall and become a part of the cell's genome. And this would affect the transformation efficiency in the end, because if, for example, the CaCl2 salt that we used made it easier for the plasmid to make it through the cell wall and the magnesium chloride did not, then that would make the transformation efficiency much lower in the magnesium chloride cells. So it would make it much harder for the cells to transform, or they wouldn't transform at all. And then our hypothesis is that the magnesium chloride will have similar effects on the cell's membrane that will allow the plasmids to make it into the cell wall.
So for our results here, you can see that this table here was taken from the lab. Our cells that were treated with the calcium chloride grew 204 colonies, and that was in our 1:10 dilution series plate. And then over here you can see our cells treated with magnesium chloride grew 85 colonies, also in our 1:10 dilution series plate. On this slide here, you can see we calculated the normal transformation efficiency and the variable transformation efficiency. So for our normal transformation efficiency, we got 4.08 times 10 to the fifth colonies per microgram per milliliter. And then for our variable transformation efficiency, we came out to a final number of 1.70 times 10 to the fifth colonies per microgram per milliliter. And you can kind of see pictures of those right here that I included here on the slides of our plates. And then for the full difference, we calculated a final number of negative 0.5833, which is less than negative 0.5.
All right, so a conclusion that can be made about our data was that the transformation efficiency of the calcium chloride treated cells was quite a bit larger than the transformation efficiency of the magnesium chloride treated cells, as the transformation efficiency for the CaCl2 was 4.08 times 10 to the fifth colony per microgram per milliliter, and the magnesium chloride treated cells was the 1.70 times 10 to the fifth colonies per microgram per milliliter. And like Jimmy said, the full difference indicated that there was a significant difference in the transformation efficiency of the magnesium chloride treated cells and the calcium chloride treated cells. And that full difference was negative 0.5883, which was less than the negative 0.5. And so based on that data, we were able to conclude that our hypothesis was incorrect, since the magnesium chloride treated cells had a decreased transformation efficiency than the cells treated with the calcium chloride. And here are some of the references that we used to come up with our hypothesis.